Great. Thank you so much for joining us today on our Career Conversations podcast. And we're very excited to be joined today by Dr. Susan Wente, who is a Beckman Young investigator and has risen now to be president of Wake Forest University and is one of our newest board members at the Arnold and Mabel Beckman Foundation. So Dr. Wente, thank you so much for joining us today. And we're really excited to hear about your career path and any messages you have for those who are considering what their own career might look like. So just to kind of get us kicked off, I would like to start at sort of the beginning. What was the first thing you did in science where you said, this is really going to be for me, I'm really excited? And when did that happen for you?
Well, I really appreciate this opportunity to share some parts of my experiences to help others. So thank you for giving me this opportunity. And I always say that, I really discovered my passion for science as an undergraduate. And I was a sophomore in college, and I was trying to decide on what my major would be. And I would say it was both a person and an opportunity. I remember really clearly my first meeting with Dr. Jean Latta, who was the undergraduate major advisor in biochemistry at the University of Iowa. And he laid out for me all the interesting aspects of biochemistry. For a Bachelor of Science major, you actually were required to do research in a lab. But he also took a real personal interest in terms of, during our conversation, he discovered that I was helping fund my education by working in the dormitory dining halls. And he helped me find a really great job in the lipid core lab, where I did my first hands-on experiments purifying and assaying enzymes, and I was hooked. That's all it took.
That's amazing that they saw you and pulled you out. So you were hooked. And what kind of research did you do as an undergrad?
As an undergrad, I worked in that lipid core lab for a couple of years, but then I also then did honors research with Professor Alice Fulton, where I was trying to purify an actin-like protein from bacteria and was very focused on protein chemistry. So that's my honors thesis, was on a lot of failed attempts to do that. It taught me early resilience in that regard. But I also, years later, when actually there was an actin-like protein purified from bacteria, I was like, yes, it did exist, right?
It's worth it.
That's right.
So did you carry that forward then in grad school?
Right. So in grad school, inspired by both Alice Fulton and also another faculty member, Peter Rubenstein, I applied to graduate school and I went to UC Berkeley to get my PhD in biochemistry. And I was focused on proteins. I went to Berkeley because of the amazing faculty there working on protein biophysics, protein biochemistry. And after doing rotations through different labs in those areas, I joined Howard Schachman's lab to work on aspartate transcarbamylase, and openly embraced every biophysical technique you could to really understand ATCase's allosteric mechanism and its structure and function.
Yeah, so I'm kind of interested in something you just said there, where you did some rotations through people's labs when you started grad school. Can you talk a little bit about what that looked like, and how did you then settle in on the lab that you decided to join?
Right. So every biochemistry graduate student had to do 3 different, visits to 3 different labs. I don't know if they were 6 or 8 weeks long, so that by March, you were making a decision about which lab to join. And all 3 of the laboratories I did rotations through, Howard Schachman's, of course, Dan Koshland, at the time another protein biochemist, and Jack Kirsch, an enzymologist. And I was drawn to Schachman's lab really because of the types of questions and approaches that were being used. And really, Schachman himself, an outstanding mentor and an outstanding teacher in regards to where I thought I'd really get a great opportunity to immerse myself in learning.
So out of grad school then, were you just thinking, I'm going to start my own lab?
Well, no, postdocs were the tradition then too. What I did learn in terms of studying ATCase, even though we were generating site-directed mutations and purifying from cells, we were really focused on understanding that enzyme in a test tube. And through other kind of really dialogues and discussions with other faculty members at Berkeley, I wanted to make a transition to not just studying proteins in a test tube, but also thinking about their functions and properties in cells.
Mhm.
And so I first made my first postdoc was studying the insulin receptor, because I'm like, everybody cares about what the insulin receptor does, right? It's an incredibly important in cell physiology. And I worked with Ora Rosen at Memorial Sloan Kettering Cancer Center for a little over a year. And then due to her passing, made a move across the street to Rockefeller University to work with Gunter Blobel. And that's really where I took that opportunity to learn about cell biology, to learn and think about protein function within cells. And that started what became my career's work on the nuclear pore complex and nucleocytoplasmic transport.
So you became a Beckman Young investigator.
Yes.
How did you hear about that opportunity? What drew you to apply for that program?
Well, I was at Washington University in St. Louis as an assistant professor. And of course, you were always looking for every grant opportunity possible. And so I wrote for the fall of 94 deadline, or fall of 95 deadline. Which would have it been? Fall of 95 deadline. I was going to say I remember it really well because I had a daughter born in July of 95. And it's true because I was a Beckman Young investigator in 96.
Wow.
So I'd been a junior faculty member for about 2 years when I started writing that grant. And it really, the description of it in terms of really trying to apply new strategies and new approaches and to develop new technology to answer, long-standing, chemistry or biochemistry problems, really resonated with some of the ideas that I was having about how to understand the mechanism of nuclear pore complex assembly.
Yeah, so that must have been a very interesting time of your life, new baby, new grant opportunity now. Can you talk a little bit about how you handled all of that?
Yeah, moving to a new place, starting your lab, starting your family, applying for grants. I was really fortunate to have a very supportive department environment. And
Mhm.
that was also something I took into account when I was thinking about positions and where to go. And, the fellow faculty members were very, I think not only experimentally, but also in other ways, mentors. Now, I was one of the first two women hired in that department, and
Okay.
the first preceded me by 3 months.
Mhm.
But we were, very much part
Well,
of
yeah,
kind
you've
of
got a
that.
group. Yes.
That's right. We were part of that new generation, and I think one, I have an incredibly supportive husband. It's actually our wedding anniversary today. We've been married 35 years.
Wow.
So, and just we were truly partners in terms of thinking about that work-life balance. And then WashU had a childcare, that had available open spots for infants. So that helped a lot too in terms of thinking about that work-life balance.
Yeah. So, when did you kind of begin to move into administration? So you're junior faculty, you have a robust research program going. What drew you to look at, and I don't know what your next step was, department
Yeah.
chair, or what did you do?
Well, I think, what has been true across many different time points in my career, whether that was when I was an undergraduate or a graduate student or a postdoc or even assistant professor, I would often get drawn into like organizing for, whether it was organizing retreats or whether it was organizing, first year curricula for graduate students, or whether it was leading other types of initiatives. I found myself drawn to really playing a role in how to make our lab or our department, more successful in terms of being places where others could discover and could thrive. And so, and sometimes, when you show a proclivity towards being an organizer and a leader, you get asked to do more and more. And so I found myself even at WashU as an assistant professor and then a tenured associate professor, taking on more and more leadership roles. And my department chair, Phil Stahl, also offered me leadership opportunities through introductions in, national organizations, the American Society for Cell Biologists, for example. So I was really, thinking about how can I have an impact that, was both for my lab itself, having a strong department is important for your own laboratory, but also in terms of how I could help others be successful too. I would say my first real recognized administrative role was as a department chair. And so I was recruited in 2002. So that was 9 years after I had started at WashU to Vanderbilt University to be chair of the cell and developmental biology department. And that was, really a significant step in terms of taking on administrative leadership. I think department chairs have some of the hardest jobs because, they're on the ground in terms of working with students and faculty directly, but then also working to try to help the organization achieve, and the school to help achieve its goals too. So I think I was drawn to it by trying to really, as I said, think about how we all work better together. And I love mentoring, not only my own students, but also that draw to be a department chairs, because mentoring junior faculty as an example.
So was this a really intentional thing that you did, or you showed up and then you got more assignments and
Right. I would say I was not looking for department chair positions at that time. And I do know when I was reached out to and asked if I would, submit my resume to be considered, I said, no, no, no. I've got a 3-year-old and a 6-year-old, and, everything is great here. My husband and I's labs are flourishing. Come back in 5 years. Right. And the person who was calling me to kind of get me interested in the position had previously been at WashU and had been recruited to Vanderbilt to be chair of pediatrics, Arnie Strauss. And Arnie said, hey, you've never been to Vanderbilt. Just look at this as like a seminar trip. Go down there, talk to them about your research. It's a low risk and, endeavor. And so I said, okay, I knew Arnie, I trusted him. And so I went down and gave a seminar, and then I started dreaming and thinking about, how this cell biology department could become a department of cell and developmental biology and really do something unique and impactful. And those opportunities don't come often, is what I also learned. So it might not have been totally the right time, but it was the right place. And that really gave me, it really energized me.
do you have any thing that you feel like you really didn't handle well, or any regrets where, kind of thinking back how you might have made that decision differently? Or
Well, I do know that I think I tried to take into account every single aspect of, that transition in terms of negotiating. And I think what you learn afterwards is that there is never a final negotiation. You are always advocating and always, um, trying to find new approaches or new ways to accomplish what you think your department or the group that you're leading needs. So that was a real lesson learned. You can try to think you've done everything on the front end, but you have to really be prepared to be continually advocating and leading.
and then at Vanderbilt, you moved into higher levels of administration. Was that all? Now that you're department chair, are you thinking, oh, this is where I belong within the university structure?
Well, I loved being a department chair. It was great. I recruited 13 faculty over 7 years. We really had an amazing graduate program. And I wasn't looking for opportunities, but there was a transition in the medical school and medical center's leadership where the new dean was going to be appointing an associate vice chancellor for research and a senior associate dean for biomedical sciences. And he approached me about taking on that role. And again, I got excited about how to have another level of impact in terms of at the institution. And now I'd be helping not just one department, but I'd be helping, all of our departments and all of our graduate programs. And so again, I could say, I hate this adage, but it's right place, right time. And certainly I could have said no, but And I don't want to say that, I felt like I'd accomplished a lot in those 7 years. And so was it the right time also for the department to, get new leadership in terms of taking it to the next level too?
Yeah. So how would you advise someone who's thinking, I like also this,
Mhm.
what you're saying about administration and helping and organizing and bringing the whole group together? Like, how do you get the skills you need?
Right. Well, there are both the passive ways, right? You have lots of role models. You kind of observe others and think and learn from both their successes and their challenges. And then there are also along the way more formal leadership development activities. And I actually was very fortunate to have access to many of those at Vanderbilt University. The dean of the medical school was very proactive about leadership development in terms of both curricula as well as group and retreat activities to help you really recognize what are things you need to strengthen or things you might need to build as capabilities and capacities as a leader. I think, my advice to others who are thinking about this is take some opportunities to do some of the smaller roles. See if you really enjoy it. It
Mhm.
has to ignite and fill some aspect of passion for you because it does involve a trade-off. Now, I ran my research lab the entire time I was an administrator up until I moved to Wake Forest. So after I was Associate Vice Chancellor for Research and Senior Associate Dean for Biomedical Sciences for 5 years, I then applied for the open provost position at Vanderbilt University and was selected for that role. And I changed how I approached my research lab. In other words, I stopped taking graduate students and refocused on postdocs and um, you know uh, research track faculty so that I would have a strong cohort. I still welcomed undergraduates into my lab, but I felt very carefully about how my research team was going to be supported. At the same time, I was involved in administration. So you do, if you're a scientist thinking about administration, you have to think about how am I going to navigate those choices and do it in a way that I would always say do no harm, right? In regards to both your own research as well as your administration.
So now you've transitioned to Wake Forest, so you're just sort of you're out of the research lab now. And
Yes,
do you have any regrets? Do you have
I
stuff
am somebody who has never looked back at any
like
step
that?
I've ever taken. I've never looked back because I said I loved being a department chair. I've loved every single role and position that I've had for a variety of different reasons. And I now truly live vicariously, right? I live vicariously by talking to our students and our faculty who are involved in any type of research or scholarship project. So here at Wake Forest, I have the great fortune of having, really strong science departments and incredible, students and faculty who are working on projects. So I can go over and, soak it in anytime I want.
How do you find mentors now at the level that you, even at a provost level or as a
Yeah.
university president? Where do
Mhm.
you go to seek that support from?
Right. There's, wonderful peers network that I have across the country in terms of, so I think, there's peer mentoring that happens. There are many presidents and chancellors of universities now who I've known along the way, for example, who were provosts when I was a provost. So that cohort, remaining very supportive of each other and sharing best practices. And then there are also retired cohorts of presidents and chancellors who serve either informally or formally as coaches for those of us who are making our way along this career path. So it can be lonely in any of these leadership roles. I don't want to claim that being president is the loneliest, because I think in any leadership role you have to think about who are your mentors, who's your support network, and be reaching out for those connections always.
Yeah. Well, thank you so much for taking us on a quick trip through a very long career. I appreciate hearing your little nuggets along the way. Is there anything else that you would like to say, that just for those starting out, maybe considering this as a career opportunity, any advice you would have given yourself?
Oh, I don't know, advice. I give myself lots of different advice. I said one of these in terms of, build a supportive network. And I think, always be building your confidence in terms of aiming high. Don't, underestimate the impact that you can have. And I think I also said, this lead in the small moments as well as the big moments is
Mhm.
incredibly important.
Great. Well, thank you so much for your time today. We really appreciate you joining us.
Well, thank you so much, Anne. It was wonderful to have this time to talk with you.